בעצם כן מכירה את כולם, וזה פחות עליב אותי, אבל...
איך מכירה את כולם?
כי אנחנו רק 8, וכאילו כולם במסדרון שלנו בערך. עוד 2 במסדרונות אחרים.
יופי, זה לא במסדרון שלי.
כן. אבל אני חושבת שכזה... כן, אבל ההתלהבות מכזה ליצור מפגש כזה נוצרה בעקבות זה שפגשתי אותך, ואני כזה, אומייגאד, יש עוד מלא שחף שאני לא מכירה.
רגע שכולם שחף. לא, את צודקת, אבל... וואי, הייתה שתסתכלתי כאילו בתאריך, כאילו, בסדר, אני יכולה ב-24, חוץ מכל הדברים, אבל נגיד ב-16 לא, ואני רואה שאף אחד מאיתנו לא מסוכן. שזה גם קצת...
זהו, הקטע אבל שיש רק 2 תאריכים, כי...
זה הסיבות שלה.
כי יש מישהי שקוראים נועה, שהיא הייתה בעסק. זהו, היא הייתה בעסק, והיא פרשה...
הבנתי.
אז היא צריכה להשלים תואר שני, בגלל שדחו את הסמינריון לשנה הבאה, אז היא כאילו נפלה כזה בין הכיסאות. אז עכשיו אנחנו מארגנים את זה, כי היא הייתה בעסק, היא לא הייתה תואר שני.
נכון, אבל השנה היא נפלה בעסק? כאילו ממש השנה?
כן. כאילו היא סיימה את השנתיים?
כן, עכשיו הבנתי.
כן, אבל אנחנו די עושים את המפגש הזה בשביל שהיא תציג ויתנו לציון.
כן.
אז אולי זה לא הכי קריטי שכולם יהיו.
כן, לא, אבל חשוב יותר מקסימום, אני אראה שאני עושה את זה ב-16.
זהו, כנראה שזה יהיה ב-16, כי יש יותר אנשים שזה יהיה ב-16.
לא, עכשיו ראיתי כזה טוב, בסדר, נראה, כאילו זה פה, זה קל. יש פה 6 ופה 6.
אוקיי.
נראה כזה.
תגידי, את יכולה לראות את המיילים של אנשים שהצביעו? כי יש פה נועה שהצביעה.
לא.
כי היא נתנה לי את התאריכים האלה, 16 ו-24, ואז פתאום אני רואה שיש נועה שהצביעה. כן.
יכול להיות שיש עוד נועה.
יכול להיות, אבל לא זכור לי שיש עוד נועה.
יש את הרשימה שלהם?
לא. לא, כי זה נשלח כזה ברשימת תפוצה של המזכירות.
כן, נכון, אבל אי אפשר לראות למי יש לך?
לא.
נכון,
מה הסיפור של השיעור שאני אומרת לך?
מי זה היה מודע?
לא. כאילו הם אמרו שזה נדחה בחצי שעה, זה נשמע לי רשמי כזה, לא קיבלת את זה.
זה ממש לא רשמי, מבלבות מבער.
התגובה ואמרה? אוקיי.
זה היה כזה.
הבנתי.
דווקא הכי לא רשמי.
טוב, החלטתי בכל מקרה, זה בא למצוא.
כן, כן, זה מאוד טוב. זה לא שקילה עם 16 מוכרח לזה. שזה היה עם כל הראשון החמישי. סתם אני לא באה.
נכון, למה?
כי אני עובדת גם בוויצמן וגם פה.
בוויצמן?
כן. כי אני יצאה מסכימה על מרכזי שהם אוטיסטים, וכאילו הם לא יכולים להגיע לפה. כאילו, אז היה לי בירוקרטיה עם ויצמן, והיא צריכה ללכת לשם ללכת, כאילו, כאילו, מה זה הייתה? מזכירות שהן כנראה, היה ממש בלגנטי, כל פעם זה איך היא עשתה, במקריות, זה ראשון וחמישי. זאת יכולה לפגשתי בראשון, בשנייה יכולה לפגשתי בחמישי. עכשיו זה לא דברים שכאילו בא לי כבר להריץ את זה. כאילו, אמרתי טוב, יאללה.
כן.
אין מה לעשות. זהו, עכשיו חצי שבוע שלי שם, חצי שבוע שלי פה. כאילו זה בכלל יוצא לי מצורף.
ואת גרה בירושלים, נכון?
לא. בבית.
וואלה. אוקיי. אז טוב, כאילו זה תיק להגיע לפה בעצם. לוויצמן זה יותר סבבה.
כן, בוויצמן אני באת לרכבת אחת ומגיעה. לא, אבל זה לא העניין, העניין זה שסתם נתחלת את זה לכל השבוע שלי, עושים לי פה שאני כאילו...
וואי.
זה ממש מפסידה מה זה דברים.
איזה קטע שגרה בהרצליה?
כן.
גם תואר ראשון בהרצליה?
תואר ראשון שלי בהרצליה.
אוקיי.
בכלל לא בבנצליה.
אוקיי.
ואז הגעתי לפה. אחרת לי לומד באוניברסיטת תל אביב. כאילו, משם. לומד כזה תואר ראשון והנדסה, כאילו, הרבה יותר.
אוקיי. טוב, כאילו מצד אחד תואר ראשון זה באמת לא כמו תואר שני, אני חושבת שאני עבור בשבוע פה, לא?
נכון, כן, לא. צודקת, אבל כאילו אני הרבה יותר פרילנס, אני הרבה יותר כאילו יכולה להגיע הוא כאילו 8 בוקר, כאילו אני לא בירושלים, אז לא היה כאילו הרבה יותר כאילו מה לי. כן.
הבנתי.
זה מה שחרץ,
זהו, אני כאילו בתפיסה, אז הייתה מישהי שכזה מפסיכוביולוגיה, שכזה אמרה לך שלום ונורא התלהבה, אז פתאום כאילו אולי...
כי סיימתי ביולוגיה. אני עברה כאילו רק
אז עשית שנה של השלמות?
כן.
מה השנה?
שנה חופשית כזו. גם אפשר סבבה. כאילו רק עבדתי ולא כסף.
אוקיי.
סיימתי כזה מלא קורסים שלהם. נראה לי זה 7 קורסים שלהם.
בגבעת רם?
כן. זה בסיס כאלה. בסיס, שזה ידעתי. אבל רק מהגנטיקה אני עוד לא ידעתי. גם לא מתעסקת בזה היום. אני חושבת את כל מעניין של זה. זהו, כן. אני ממש ראיתי שאני אוהבת מאוד להתעסקות מזגת.
זה מגניב, כאילו אני כל הזמן חושבת על זה שבאמת למי שזה מעניין אז אפשר לעשות דברים ממש חשובים.
ממש חשובים, כן. אני מסיימת לב מזה. אבל כאילו בגלל שהגעתי לפסיכולוגיה, הלכתי למראה, כאילו כזה.
אז למה היא צריכה להשלים ביולוגיה כדי להתחיל במשהו מרמות?
כן, כאילו אמרו לי שכזה אני צריכה להשלים, כאילו בעיקרון, בואי. פסיכולוגיה למדתי את רוב הדברים שאני כאילו צריכה כדי ללמוד פה. אמרו לי כאילו לא, את צריכה לעשות ביולוגית, לא למדתי בתחום ביולוגיה, למדתי בתחום פסיכולוגיה. שאולי כדי שאני חושבת שזה תרם משהו, לא. זה הייתה עוד שנה כזה של לבדוק את הדברים, והתחלתי במעבדה של מירב לפני. בגלל זה השנה התחלתי מוקדם את התזה שלי, אני הלכתי את רוב כאילו, אין תרם לזה.
כן, כן. טוב, מגניב. יופי, גיליתי דברים. לא, באמת הייתי לעצמי איפה את, ולמה את לא מגיעה. חשבתי שאולי את מעדיפה כזה לראות את השיעורים.
זהו, שאין את השיעורים בנאום זה.
לא אין?
שאר החוזים שאני מקודן כזה, גם לא כאלה פעילים. גם שלי שנה שפרסמים שנה שעברה.
זהו, בסוף יש.
כן, לא מצאתי אותו בכלל.
יש להם פיקיה של אפס.
יש לי, כן, זהו.
יש לך פגישה?
אז לא, אז פשוט מה שהיא תשלח לי את זה. זה אני מספיק בסדר שהיא אמרה את זה. תגידי איך הולך לך את זה?
סבבה,
שאלת משהו?
כן, היינו באתיופיה לפני שבועיים. חזרנו לפני שבוע. היית כשחזרת?
כן.
זהו, אז הוא חזר ואנחנו עדיין היינו שם.
ואתם עדיין הייתם? מגניב. איך היה?
היה ממש טוב.
חוויה גם, נכון?
ממש, ממש ממש.
כן, גם טורפת.
כן, כאילו קשה כזה בדיוק להסביר מה היה שם, אבל זה ממש, כן.
זה באמת חוויה, אבל לא כאילו.
כן, כאילו אפשר להגיד, היה מושלם.
חוויה.
כן, כן, אבל לא, אבל זה היה משמעותי, כי הם ילדים מקסימים, אני כאילו בחיים שלי, עזבי ילדים, לא פגשתי אנשים כאלה מסוקים.
כן.
יש משהו אולי בעיזרון, אני לא יודעת, כאילו גורם לאנשים להיות כזה קצת מסוימים, אבל גם נורא נורא מתוקים.
אני חושבת על זה, בשיחות זה. ילדים וזה, והיא כאילו אמרה אין אני לא צריך להבין שזה ילדים אחרים. היא אמרה לי, ילדים
למרות שהיו לנו קונטרולות בבית ספר כנראה, כאילו לפני והם היו ילדים כמו פה.
כן,
זה כאילו לדעת לעשות את זה.
אני כאילו, גם אותה, גם
טוב. מתנצל על האיחור. שכחתי את המחשב שלי בהדסה. צריך להביא אותו. ועכשיו ייקח לו קצת זמן להתעורר. נתחיל בלי. זהו, עוד אין פה כלום לראות.
טוב, אנחנו רוצים שתקליט את עצמך.
מישהו הביא את זה? כן, אפשר לדעת. זה דולק.
מה זה שדולק?
הדלת של הפודיום צריכה לפתוח.
מימין, זה פתוח.
היא פתוחה איתך.
כן. אז מה, לשים את זה?
יש
מה
פה.
זה זה? אתם יכולים לעשות את זה?
מיקרופון?
כן, זה. טוב, אתה משנה את זה.
זה בהתחלה.
זה נקרא.
זה בהתחלה.
זה גם אמור להגביר אותי? אתם תדעו אם זה עובד?
רק אם ממש תתקרב זה...
כן, אז בהתחלה הם לעשות שוק קטן מאוד.
חוץ מזה ששכחתי את המחשב בהדסה, המחשב שלי עכשיו, כל פעם שהוא מתעורר 10 דקות הוא לא מדבר. אז אנחנו 5-10 דקות ראשונות נהיה בלי, ואז אני אפתח לכם גם משהו לראות. אז אני דני רוקני, אני English? Who needs English? Yeah, okay. So, my name is Danny Rockney. I'm a member of the Department of Neurobiology at the medical school, and I study the olfactory system. And I'm going to teach you about the physiology of the olfactory system. So, like any other sensory system, the first thing you want to know when you start studying a sensory system is what the stimulus is, right? How do you describe the stimulus? And for, I'm going to go back and forth to see if my computer is willing to show the slides. So for vision, we have a metric for stimuli. We have different metrics for different features of the stimulus, but one of them would be wavelength, right? The color. So you have a scale, and we have wavelength. And there's a certain range that we are either sensitive to, and we can say that two colors are similar to each other just because they're close to each other on this axis, right? If you want to take a different feature, like the orientation of a line, then you have the angles define how similar two lines are, two orientations are. Same for sounds, right? We have a metric that can describe how similar two sounds are relative to each other, right? Where they are in stimulus space. Okay, maybe you can see the slides now. No, not yet. So what are the stimuli for the olfactory system? What are they? Chemicals, right? And then that's up. And I need to turn something on here?
Yes,
Projector? laptop HDMI. And the screen, how did the screen come down? Okay. So this you already know, right? Okay, so we said that the stimuli for the olfactory system are different chemicals. So how do we define the space, right? What are the metrics that we use to describe different chemicals? Does anyone have any idea? What would be the equivalent of wavelength in chemical structure? Concentration. So that is one variable that changes kind of continuously. So we have many, many attributes to describe chemicals. None of them are actually providing a metric. Okay? So one of the main difficulties in studying the olfactory system is that we have no way of varying a stimulus going from one chemical to a different chemical in a gradual way. Like you would do with, you can easily go from orange to blue through all of the wavelengths, right? There is no way of doing this in chemical structure. And not only that we cannot vary it continuously, we don't even have good definitions of what similar molecules mean. Okay, and I'll get back to this in a second. I skipped one other thing that I want to tell you about the actual biology of the system. That is that there is not one olfactory system. There are actually several. And together we think of them as chemical senses. Okay? So one of them, and that's the one I'm going to talk about today, is the main olfactory system. This is your nose, right? And everything in the brain that gets input from your nose. Others are what we call accessory olfactory systems. Some of these we don't have, we as humans, but most other animals do. And these are thought to be more specialized. Okay, so this is thought to be a general system, taking kind of any input, making sense out of it. These are thought to be specialized for pheromones or cross-species inputs, so detecting your predator, stuff like that. And obviously we have taste, so that's another chemical sense. And these are somewhat similar. They all are activated by chemicals, but they have different sensory organs and different, at least peripheral brain areas that deal with them. Right? If we go deep enough, then everything converges anyhow. Okay, so we're going to talk about this system. And I think you have a couple of hours with Yoram after me who's going to talk about these systems. Okay, so going back to the stimulus. So first of all, what do you need smell for? So for humans, our intuitions are not very strong for what olfaction is used for in nature. And the reason is that we use vision for many things that other animals use olfaction for. But most mammals, certainly rodents that we tend to study a lot, use olfaction for all of these things. So this is the main sense they use for social interactions, for finding their food that is often not visible, so either underground or for many animals in the dark, and also detecting predators and avoiding them. So if the mouse sees the cat, it's kind of late. If the mouse smells that there's cat urine in the area, then he can already change his behavior to avoid the predator. Now, this is not saying that humans don't need olfaction. We obviously use it for caring for offspring. We assess the quality of food based on olfaction, and you can also identify some dangers based on olfaction. And some people claim that these are all kind of extreme cases. Some people claim that we have a lot of unaware decisions that we make based on olfactory cues. How strong is olfaction for a rodent, for instance? So one nice experiment that people have done multiple times by now is you take a male mouse, and he has its own cage. And now you introduce a new male mouse. So this is an intruder. And the original mouse would attack this intruder. And now you take instead of the wild type mouse, you take the homeowner is now some mouse in which some part of the olfactory system doesn't work properly. And people have done this with either the main olfactory system or the accessory olfactory system. And now you introduce the intruder. What happens? Has anyone ever seen this movie? No. He's trying to mount the intruder, okay, instead of attacking him. So any change in the olfactory system that people have tried, which are 3 different changes that I know of, have resulted in the same exact result. Okay, so it's a very crucial input for them to make decisions about at least social interactions. Okay, so going back to the stimulus. So I already told you that we have this problem of metric. Now, what are the things that we actually smell? So if you look at an object that we can identify by smell, bread is one of them, then you'll find that this object emits a lot of volatile chemicals. Okay, so in order to smell anything, we need the chemical to be volatile. If it's very heavy and goes down to the ground, we won't smell it, right? It needs to be in the air. You take the bread and you look. There are. Does it have a number here? No. So typical objects are from tens to thousands of volatiles that we can actually detect, each one of them individually. But what makes the object for us is the specific mixture, right? So any object is many of these chemicals. Coffee, another one. Some of these objects you can mimic with just a single chemical. So for instance, banana emits a number of chemicals that together make us feel the smell of banana, but we can make a molecule that is called isoamyl acetate that smells really similar to banana. Okay, so there is some kind of unpredictable nature where we don't understand why this chemical smells like this mixture. Okay, the stimulus itself is a big mess. Okay. So going back to the metric. So there are different questions we would want to know about the odor world, right? First of all, what can we smell? Probably not all molecules in the world, right? So if they're not volatile, we don't smell them. But can we smell all volatile chemicals? We know that the answer is no, right? How do you know the answer is no? There's one dangerous chemical, gas, right? So the gas at home, the reason you smell it is because we add something smelly to it so that you know if you're in danger. But we don't smell the gas itself. So not all molecules. Which molecules? We don't have a rule that we can say, we can't predict, right? Take an unknown molecule and predict whether we can smell it or not. We don't know how to predict that. How many different smells can we smell? That's a very kind of important question. Not the exact number, but the range. Is it hundreds? Is it billions? How many are there that we can actually smell? We don't know the answer to this. There is one recent attempt. I don't know if anyone saw it in, I think 2015. Science, there was a paper saying that we can smell at least 1 trillion different smells. And then, obviously, you can't do the experiment, right? You can't take a person and let it smell 1 trillion different odors. So you need to extrapolate. How do you extrapolate? If I draw here, will you see it? Yeah. So the same question was asked for vision many years ago. How many colors can you see? Does anyone know the number? It's around 1 million. How do you know? So we know the range, right? So this is wavelength again. We know the range, right? And we can define the resolution. I can just give you two similar colors and ask, can you still tell them apart? And then find what's the minimal difference that you can tell them apart. And then just divide the range by this minimal difference, and you get 1 million.
But it's not, we don't know for sure that in all the spectrum it's the same.
True. True. So you can try this around here, and around here, and get a number. You know, if it's 500,000, it's fine. But it's not 100. So people have tried doing something along these lines for olfaction and got the number of 1 trillion, and everyone was very excited, including Haaretz's paper. And then there was a flaw, and the main flaw is that we don't have a metric. So how can you do this? How can you extrapolate? And people have redone the math on the same data that they used, and the number they got to is 10, at least 10. So anywhere between 10 and 1 trillion is probably true. So we don't know the answer. Okay, and so really this is really the difficult question. I already started talking about how do we define odor similarity? How do we define what the resolution is? Right? And here are just some drawings of chemicals that we can smell. All of these would smell, if you'd smell them, you'd probably use words something like fruity, flowery, perfumey, something like that, okay? But which two are the most similar, right? How would you rank them? You can use some intuitive measure and claim that maybe these two are pretty similar, but are they more similar than these two? How would you put numbers on these and compare? So there has been there have been a few attempts, but I'm just going to describe one attempt that has been made by mostly by Noam Sobel from the Weizmann Institute. And the idea was, what if we take the features that were mentioned here a while ago? So you can take a chemical and go to a chemist and get a list of features, right? So you have the molecular weight, the number of carbon atoms, the compactness of the molecule. You can get, there are about, I think, over 1,000 different features that you can define for a molecule and get a number for each one. So the molecular weight is going to be going to have a value, right? Just take all the values and now do that for many, many chemicals. And now what you can ask is, okay, if I plot, let's say I'm now looking at just the two features, okay, just these two. If I just take each dot now as an odor, right, a chemical, and it has the value of this feature and the value of this feature, okay? And what you can see here is that these features are somehow correlated, right? There's a correlation between these features. So I really don't need the information of both features because they're really very correlated. If I just use the right axes, let's say transform the axes this way, then this axis kind of gives you most of the information, right? And I'm not sure if you've already learned this or not. This is called principal component analysis, the way they did this calculation, okay? So they did this, but not for these two features, but for all 1,500 or something different features. And basically you ask, are there really strong correlations? Is the space really that large? We need 1,500 different features to describe chemicals, or can we use much less? And the answer was that you can actually use much less. So this plot, please stop me if anything is not clear, okay? This plot shows how much of the information you explain in each of these axes that you pick. Okay, so PCA, you haven't learned PCA, right? Or you have? No. So I'm not going to explain it, but just the gist is that PCA finds kind of the first axis is going to be just the best axis, the one that gives you the most information. And the second is going to give you the second most information. There are some rules which axes are allowed or not. You need, they must be orthogonal. But so the point is that you kind of rank order the axes that you choose based on how much information they give you. So the ones, the 1000th axis may have almost no information. You don't need it. Right? You can just use, in this case, you can just use one and you're pretty happy. What they found is that if you just use one of the 1500, you already get about 30% of the information. Okay? The second gives you another about 10%, right? And then it drops. So if you're willing to use 10 of these axes, you get to almost 70% of the information. So their point was that even though chemical space sounds like really badly defined and hard to work with multidimensional space, it is multidimensional, but maybe not the number of dimensions, maybe much less than we initially thought. Okay, so this is a chemical analysis. But what they went on and did is a very similar analysis, but now use perceptual features. So basically, they took the same chemicals and let people smell them and asked them to just rank, right, on a number from, forget, 1 to 10 or 1 to 5, whatever. How flowery is this smell, right? And then how sweet, how orange, how almond. And they had a long list of perceptual features. You take multiple people, let them smell it and rank it. They chose specific people who work in smell industries, either perfume, wine, food. And then you can do the same thing. So you average across all the people, hoping that this keeps in, the amount of information that you care about. And now you can do the same thing. You plot here now perceptual features. Okay, so let's say this is how flowery each odor is, and this is how sweet it is. And each dot is one odor. Okay? And again, they're correlated. This is a cartoon, right? This is not real data. And then you can do the same trick again in PCA, rotating the axes to find how many features you really need to describe the percept. And the result was very similar. You need pretty much the same amount of features to get to the same level of information. Okay, so their claim is that both at the chemical level and at the perceptual level, space can be better defined than just using thousands or hundreds of features. You don't need that much if you choose the right ones. The right ones, by the way, are not easily always defined. Okay, so if this is flowery and this is sweet, what is this axis? Labeling it with a name is not trivial, but maybe it doesn't matter either. They did, by the way. They claim that the first axis is pleasantness. Okay. Okay. So this is kind of the first attempt. It's not the best so far, but it's probably the one that made the biggest step. Since then, there have been some improvements of how much you can predict the percept based on the chemicals that you put in. But this is still extremely noisy. So basically, So where we are at this point is that people keep making these attempts, which is kind of heroic. This is really difficult, and these are really large groups. I just put these here. If anyone wants to look them up, you can later. But basically, no one will claim that, give me a chemical, I'll tell you what it smells like. We're not there. Okay, so this is, I would say, the biggest difficulty studying olfaction. We don't have a good understanding of the stimulus itself. Now, I didn't put a slide for it, but another issue with the stimulus is that unlike visual stimuli and auditory stimuli, even generating a stimulus in a very controlled manner is difficult. So if you want a certain contrast, you buy a really good monitor, and you can display an image in whatever contrast you like, right? Or if you want a pure tone, you can easily get a pure tone. Just buy the right equipment, and you can play, within a certain range, any pure tone you like, or any combination of tones. Way worse with olfaction. So if you want to present a certain chemical at a certain concentration, just getting to that point is going to be really difficult, and you'll also never know that you actually got there, because our tools for measuring the odors in a space, right, in an amount of air, are not very good. So that's a second difficulty. Okay, so what do we know about the biology? Let's get into the brain. We already had our break, right, before I got here, so I'm just going to keep talking. Okay, so this is a mouse head. I'm going to probably go back and forth between mouse and human, but we know way more about mouse, so most is going to be mouse. So far, most things are similar. I'll probably try to show you the differences. So this is supposed to be a cartoon of what, in general, the system looks like, the biological system looks like. So you have a nose, this is the sensor, and you have a brain, right, that processes the information. This piece of tissue here is the olfactory epithelium, so that's not brain, that's inside your nose. This is where sensory neurons lie. Okay, so these round things are supposed to be sensory neurons, and these are their axons. Axons, olfactory epithelium. The axons project to the brain. First brain region to deal with olfaction is called the olfactory bulb. So the olfactory bulb is the only region in the brain receiving information from the nose. Okay, anyone else in the brain receiving olfactory information will receive it from the olfactory bulb or second order structures. The olfactory bulb does its processing and projects to the olfactory cortex. Okay, and from there, olfactory cortex projects to pretty much anywhere you want to think about, right? Frontal cortex, hippocampus, grid cell areas. I think not the cerebellum, but most of the brain. So this is basically a cartoon, and I'm going to add some parts to it a little bit later. So I'm going to start, going step by step, and we're going to start with the sensory part. Just the human connectivity is really similar. You have the epithelium, sensory neurons lie in the epithelium, project to the olfactory bulb, which is this yellow thing. Olfactory bulb projects to the olfactory cortex, which is around here, a little bit to the side. The relative sizes of olfactory regions in the human are very different from the mouse. So the mouse olfactory regions, if you mark all the olfactory regions, it's kind of like marking all the visual regions in the human brain. You just see a lot of brain. Okay. So sensory neurons. So this is just an enlargement of this area. These are the sensory neurons. And the sensory neurons lie within the olfactory epithelium, and they have these cilia here that project into the nasal cavity. Okay? So this is where, when you breathe, this is where the air goes. So airborne odorants can come here and be sensed by these sensory neurons. Now, how do they do it? The sensory neurons, the olfactory sensory neurons, express olfactory receptor proteins, which are these 7 transmembrane domain proteins that are G protein coupled receptor. Does everyone know what a G protein coupled receptor is? Everyone. Wow. So what this means is that when this receptor is going to bind an odorant, it's not a channel. It doesn't open and let ions in. It's going to activate a G protein, and that's going to, in the end, activate a channel. Now, we don't express just one type of receptor, because otherwise how would we tell apart different odors, right? So there are different types. Each one of them tends to be activated by different odorants. And the number of receptor genes depends on the species. I'll show you it in a second. But it's the largest gene family known in the mammalian genome. Okay, it's about 3 to 5% of the genome of most mammals. And these two people here, Richard Axel, that maybe you've heard speak in the last couple of years? No? He's here every year almost. And Linda Buck, they got the Nobel Prize for identifying these genes. Okay. So the number of genes varies a lot depending on the species. And there are a lot of genetic, really interesting issues with olfactory receptors that I'm not extremely familiar with many of them, but I'll just mention. So first thing is the huge variation. So the human here has around 400. The red shows the active genes. I'll tell you what the rest is in a second. Around 400 for us, around 1,200 for mouse, rat, and about 2,000 for the elephant. Okay, so very different numbers. What's really interesting is the huge numbers of pseudogenes. What are pseudogenes? Pseudogenes are mutated genes that are not functional anymore, but are still in the genome. Okay, so all of these species have a lot. And a little bit I know about why they're there is that just the mutation, basically all these olfactory receptors tend to mutate way more than other genes in our genome. Probably the reason is that it's not that costly to lose a gene, and you might gain some new receptor. This is what people, I think about it. Okay, so this is one interesting genetic issue. How do these sensory neurons work? So first of all, where is the receptor expressed? I kind of already told you. It's expressed in the cilia. The way people have shown this is to just culture these neurons or make a preparation with epithelium. You can record from the neuron and then puff an odorant on different regions of the neuron. And you can see that only if you puff the odorant on the cilia, you get a strong electrical response. And the way we think they work, like similar to many other sensory neurons, like somatosensory neurons, is that action potentials are generated in the axon. The region that actually does the transduction is not active, so it just passively depolarizes, right? It can be huge. 50 millivolts in the soma of any neuron would generate a spike right way below 50 millivolts. But there's no spike generation mechanism here. So this travels passively to the soma. And somewhere here, probably the axon hillock, is where spikes are generated. And this is what the olfactory bulb will see in the end, right? Just the spike. Okay, is this clear? Yeah. Yeah.
So we were talking about all of these different genes that we have. So does that directly correspond to what you were talking about?
The number of...
Yeah, like how many smell, and like if it's like, okay, one gene, say, to make it easier, one gene per chemical, and then we have...
I'm going to get to that in a second. But if it was one chemical per gene, then yes, more genes, more molecules. It's not. So that's why the answer to your question is not trivial, but I'll get to that in a second. Okay. So in terms of transduction, how does it work? So if we look at one cilia here, this is your receptor. So it binds odorant molecules. It activates a G protein, specific G protein that only lives in olfactory sensory neurons. It's called G-olf. And this will activate this cascade. You don't need to know the details of the cascade, but just there are two interesting features. One, in the end, you have cyclic AMP that will activate calcium, mostly calcium, but also sodium channels. So this will depolarize the membrane. The really weird feature here is that calcium that goes in through these channels opens chloride channels. Now, normally opening chloride channels, what would that do to the membrane potential? Right. It will hyperpolarize, right? Because chloride reversal is typically around minus 60, 70. Chloride reversal in these cells is unusually elevated. So the opening chloride channels, actually the current goes the other way, not normally going in, but going out. So it further depolarizes the membrane. Okay, so there are two steps, calcium channels and chloride channels that are dependent on this calcium. So this is a cellular interesting part of olfactory transduction. Okay. So now to your question, almost to your question. So I told you there are a large number of genes, and sensory neurons express these genes, but they don't just, each neuron expresses all of these genes. Each neuron chooses one, right? So I'm going to talk about mice now because they're my favorite animal. So 1,200 genes. Each sensory neuron will choose just one, and we'll make sure it only expresses that one. Okay? Many copies, but just one gene. So so far you could have a sensory neuron being a detector for a chemical. We'll soon see why not. And again, genetically, this is very interesting because for anyone caring about epigenetics, which is the field studying which genes are expressed when, how does the system control which genes should be expressed, this is an extreme case where the one gene that is expressed somehow manages to silence all the others, right? Another 1,000 genes. So there's a few people really going into figuring out how this works. Okay. So again, looking at comparisons to other sensory systems, typically we think of the way the sensors are arranged often has some sort of map, right? What you're sensitive to depends where you're going to be. If you think of the cochlea, have you studied learned about the cochlea already? So sensory neurons, their best frequency is dependent on where they sit in the cochlea, right? The spatial location of the sensitivity in the retina depends on where you are in the retina, right? So what would be the equivalent in the olfactory system? Is there any mapping of the gene that you express onto where you're going to be in the epithelium? So these are fluorescent images. How do we turn off the lights? This one? Oh, that side. Can you see these green dots? Yeah. So this is a mouse on the left that has a green fluorescent protein in all olfactory sensory neurons. Okay, and this is the epithelium. And you just see that the whole epithelium is green, which is maybe not surprising. This is a mouse that expresses a green fluorescent protein in just one type of receptor. Okay, just one gene. It's called the I7 receptor. And you can see that it's not limited to just one spot in the epithelium, but it's also not spread over the whole epithelium, right? There's kind of a region that it likes. This is a different receptor. It has a different region that it likes. It's spread over this region, but it's still limited to some kind of region. People have done this to multiple receptors, and the image that emerges is that there are several regions. Right now, people think of 3 kind of defining regions, and each gene is kind of limited to one of these regions, but spread over the whole region. Okay? Whether this matters to anything, we don't know, in terms of functionally how you smell. Yeah. Okay. So now, is it really that one gene smells one chemical, right? And so this is cartoon data, but people have done this experiment numerous times. If you take, throughout this, in pretty much any olfactory presentation, the way you show which gene is expressed is by color. So these are sensory neurons expressing different genes. Kind of the color is supposed to tell you which gene it is, right? So this guy and this guy express the same gene. Okay, so we now take this green guy and try different chemicals on it, and you can see it responds to different chemicals, but not to all, right? Almond not, but peppermint to different extents, but responds to multiple chemicals. And also you can look at the other way. If you take one chemical, it may activate different receptors. Okay? So we don't have each gene as a detector for a specific chemical. And what we do have is really what we call a combinatorial code, in which each odorant can be identified by which genes it activated, right? Which receptor neurons it activated. So if I activated the orange and the pink, I know it's this odorant, and if it's the orange and the green, it's this odorant, right? And the way I describe it now is kind of binary, right? It's either activated or not, but we know that it's not like that. So it's not just which receptor is activated, it's also to which level. Okay? So if you think about it, if you have 1,200 different receptors, and if you think of a binary system where each receptor is either activated or not, then the possible number of combinations is 2 to the power of 1,200, which is a huge number. We don't even know if there is enough volatile molecules to fill this range, right? But you can actually do more because it's not binary. You can also look at the level to which they're expressed. So this doesn't give us any information about how many molecules we can smell, okay, or differentiate. Okay, but this is basically the way I think pretty much anyone thinks about how the olfactory epithelium encodes which odor is present. Okay, a combinatorics of the different receptors.
What you show here is not one basic chemical. It's just like another.
Oh, the word. Yeah, so you can do it with also just one basic chemical.
And also with
Yeah, same thing. Yeah. Actually, so this is from a textbook. The actual experiments were done with basic chemicals. No, people don't often use mixtures. Yeah. Okay, is this clear? Yeah. Okay, so I already told you that once you have the code in the sensory neurons, this is projected to the olfactory bulb, right? So these axons carrying the information about which one was activated and not, then is projected through these axons to the olfactory bulb. So what does the olfactory bulb look like? And then also, I'm going to just keep mentioning this. Any information going to the brain from the nose is from the olfactory bulb. Okay, there is no directly nose to cortex pathway. Okay. So this is an image of the same mouse I showed you earlier. The mouse that has the green fluorescent protein in all sensory neurons, but we're now looking at the olfactory bulb. Okay, this is the surface of the olfactory bulb. So what are we seeing here? Why do we see green? The sensory neurons are in the epithelium, right? We're not looking at the epithelium, so why do we see green? What part of the sensory neuron does get to the olfactory bulb? Axons. So these are the axons of the sensory neurons. And what you can hopefully see is that they're arranged in these circular structures, right? It's not homogeneous, but it's split into these things we call glomeruli. Okay, each one of these is a glomerulus. So this was all sensory neurons. What happens if we just stain one sensory neuron? Right? You can think of two options. One is that you'll just have a weaker signal, way less axons. But what you'd actually find is that all the axons from the sensory neurons expressing the same gene, just one gene, converge into one glomerulus. Okay? If you'd had the full image of the whole olfactory bulb, you'd find another one. So there are two per gene, typically. You can also stain a different receptor, and you'll find that its axons don't go into this glomerulus. They only go to a different one. Okay, so each glomerulus is basically the convergence of many, many axons from sensory neurons expressing the same gene, the same receptor gene. It's about 10,000. It differs depending on which gene it is, but it's on the order of 5 to 10,000 axons innervating each glomerulus.
How do they know what to convert?
Oh, that's a great question. So the receptor gene itself is part of the system, but the full picture is not known. But there are different molecules that people know guide axons. But yeah, there are two questions. One is whether, so you could think, the axons somehow aggregate and then form the glomerulus, or the axons have a target and they try to get to a specific point. Seems it's like the second version, right? So if you look in different animals, the same glomerulus is going to be in about the same position. And there are genes that you could knock out to move a glomerulus to a different location. It's still a glomerulus, it just shifted to a different location. Okay. So basically we have now an input layer into the olfactory bulb, which maintains the sensory gene identity, right? The input layer, each glomerulus still reflects the activity of just one gene, just one receptor type. Okay. So, yeah, I already told you that. So how does this look if you look now at when you present odors to this animal, what does the input actually look like? Right? What are the activity patterns that emerge when you present an odor? And what you'd find is that presenting different odors, so this is just a resting fluorescence image, and this helps you to just see the glomeruli. Okay, this is a mouse expressing a fluorescent protein. Actually, not a fluorescent protein. In this case, it's intrinsic imaging. But you can see the anatomy using fluorescence, and you can see these glomeruli. Now you present an odor, and you can see which of them are activated by the odor. Okay, and you see a pattern. And you try a different odor, and you get a different pattern. Okay? If it's not clear, then red is activated, blue is not, and then it goes through red, yellow, green. Okay, so each odor will just give you a different pattern. This is just a small part of the olfactory bulb, right? Glomeruli are all around. So different odors, you just get different input patterns that the olfactory bulb can then read. Yeah?
And the patterns are the same when you introduce over and over again?
The same odor? So, yeah, basically yes. There is some noise and variation, but basically yes. Okay, so the next question is is this a spatial code? So we get here different patterns. We see them in space, but by spatial code, I mean, does it matter that these two glomeruli are neighbors? Okay? A different code would be, we can call an identity code. So we can just, think of the combinatorics in which we just care about which glomerulus is activated, or we can think that also the position matters, right? Not just which receptor, but there's some importance in the fact that these two are neighbors. So this is, again, a question that people have been struggling with for a long time, and I'm going to kind of give you the short answer of it. And basically, there are three maps that people talk about in olfaction, and they have different names, so let's go through them. So the first one is rhinotopy. Basically, this we've already looked at. This claims that the position of a sensory neuron in the epithelium maps onto the position of where the glomerulus is going to be. Okay, so if I showed you there are three regions in the epithelium, if I know where a gene is expressed in which of these regions, can I predict where the glomerulus is going to be? Okay? This is rhinotopy. That's one map that is possible. Another one is odotopy, which really just it says space doesn't matter. Individual odors activate different parts of the olfactory bulb. Their location doesn't matter. So just the fact that you have different combinatorics is enough. And the last is chemotopy. This is the most difficult to study for reasons we've already discussed. And the idea here is that there's a systematic representation in spatial coordinates of structural features of odorants. Okay, so basically the idea is that if you have two similar chemicals, they're going to map onto nearby glomeruli. Okay, or in other words, moving from this glomerulus to this glomerulus means, let's say one less carbon atom, and then here one less carbon atom, so that you have some systematic mapping. Why is this difficult to study or even comprehend? Just because structural features of odorant molecules are something that we're struggling with to define, right? So you can come up with any number of structural features that you want to look at and look at whether they are mapped. So far people have tried tens and it doesn't seem like it's mapped. So just to give you the answers, this we are certain is true. You present different odors, you get different patterns of inputs. There's some level of predictability between where the gene is expressed in the epithelium and where the glomerulus is going to be in the bulb. Basically, I would say this just doesn't exist. Okay. Okay, so the olfactory bulb now needs to read this information. So let's see how this works. So, Matti, you promised that they are much more active. I don't know, it's maybe the fact that I was late or something. So I'm going to first describe the circuit, the olfactory bulb. What are the components there? And then we're going to see what it does with the information it receives. So this is a slice through the olfactory bulb. Okay, this down is deep. Here's the surface. And the staining is Dapi staining. Does everyone know what Dapi staining is? Matti doesn't know. Dapi staining is a staining in which you see nuclei of cells. Okay, so basically this is a map of cell density. Okay, soma density, really. So you can see, so let's go from the top. You see some cells here, and then there are these round regions that are almost empty from cells, but have a lot of cells around them. Again, an empty region. There's this one row of cells, empty region, and then a lot of cells deep. Okay? What are these circles? It already says here. These are the glomeruli. So where you get the 10,000 axons of sensory neurons converging, there's no room for many cells in there. Okay? These are the glomeruli, and then you have, let's move here. So here I'm showing the cellular components. Not all of them, but at least the, I don't want to say they're more important, but they're like classes of cells. The color codes principal neurons in green and local interneurons in blue. Okay, principal neurons, by principal neuron I mean two things. One, they receive direct input from sensory neurons, and two, they project out of the olfactory bulb, so they provide the output. They read the input, provide the output. That makes them principal neurons. Local interneurons may read the input as well, or may just get the input locally, and also their output is local in the olfactory bulb. So they'll affect the output of these principal neurons, but they don't themselves project out. Okay? These blue neurons, most of them are inhibitory, but some are not. Projection neurons are all of them excitatory. I'm going to give them names. These are mitral cells. These are considered kind of the main principal neurons. The second class is tufted cells. I don't care if you don't remember these names. And we have two classes of tufted cells. And then we have all of these neurons that lie around the glomeruli are, for that reason, called periglomerular neurons. Okay, so they mostly affect, or at least that's what we think, they affect the way principal neuron dendrites integrate the information. And deeper, we have granule cells. We only have two in the drawing, but these are the most numerous. You can see here. There are millions of them. And these mostly affect the output, okay, the spiking. What interesting features do we see here? So one thing that I should mention is that these synapses between mitral cells and granule cells are another part of olfaction weirdness. Granule cells have no axon. And this part, they receive their input mostly from this thing, which is a dendrite. Okay, so these are dendrodendritic synapses. The mitral cell dendrite releases glutamate that activates the granule cell dendrite. The granule cell dendrite releases GABA. Okay, that inhibits the mitral cell dendrite. So these are very different from the information transfer that we're used to thinking in between neurons. Okay? I'm not going to talk more about that. Okay. So this is the circuitry. This is where the information is read. Each glomerulus, I already told you, is about 10,000 axons. The number of mitral cell dendrites going into a glomerulus is not exactly known, but on average it's probably around 10, 15. Yeah?
One mitral cell gets input only for one?
Yes, that was my next sentence. Yeah. So mitral cell will send its dendrite, its apical dendrite, only to one glomerulus. Okay, so mitral cells also maintain the identity of the receptor gene. Okay? Is this clear? We'll get to that. Cortex is a mess. Okay, so this is what this whole thing we call labeled line processing, at least to some extent. Why? Because each sensory neuron is one gene, and then the glomerulus is one sensory neuron type, and then mitral cells receive information from only one glomerulus. So basically, mitral cell is mapped onto one gene. Okay? Okay. So the question is really, does a mitral cell, does its activity really reflect the activation of just one receptor gene? And why could it not be? What could allow it to not reflect just one gene? Let me go back. It's these guys. It has these very long lateral dendrites. These don't receive input from any glomerulus, but they may receive input from some local neurons that will receive input from a glomerulus. Okay, so potentially this long dendrite may allow a mitral cell to also see some other inputs beyond its glomerulus. So this is one experiment that people have done to look at to what extent the activity of a mitral cell reflects the activity of one gene. And the way they've done it, this is kind of heroic. So first you make an animal that expresses a green fluorescent protein only in one gene. Okay, so this is these guys. These are sensory neurons. They're axons. Now you can see the glomerulus. Then you have two options. One is to just blindly try to record around the glomerulus and stain the cell to make sure that the dendrite that you record from actually goes into that glomerulus. This will mean that your yield maybe is not great because you're going to record from many things that you didn't want to. Another way that people have done this is that you then electroporate DNA into the glomerulus. So dendrites that are in here may pick up this DNA, and then if you look deep, you can see neurons that express a red marker, right, that the DNA encoded for. So now you can see deep cells that have their dendrite up here. And then you can record from them. But this is just an experiment with a lot of stages that may fail. So that's why I'm saying it's kind of heroic. But what do you do with it? So here you record from a cell. You know that the dendrite goes into this glomerulus. And what they did, they used different ligands, different odors, basically, that we know to what extent they activate the sensory neurons. Okay, so they have here a list of 70 different odors. The number here is just which odor they used. And they know that this one activates this sensory neuron very strongly, and the other is not very strongly. And then they asked does the mitral cell respond similarly? Okay, so here you have 3 examples of 3 different mitral cells they recorded from, and all of them responded maximally to this specific odor. Okay, so the answer is that it at least very strongly reflects the activation of its parent glomerulus. Okay? Okay. So now these bars, I didn't really tell you what the y-axis is response strength in spikes per second, but they integrate over the same amount of time always, so you can just spike count. So this is assuming that within a range of response time, within that range, time doesn't matter, right? You just get one value of how many spikes were there. But if you actually look at the activity of mitral cells, you'll see that it comes in these little bursts, right? You see these, I don't know how easy it is to see, but each of these wider lines is just a lot of spikes, right? And why do they burst? They burst because the animal breathes. So breathing means that your input is periodic, right? It's not flat. And there are 2 reasons why olfactory neurons, if you do the same in sensory neurons, you'll see the same. There are 2 reasons why you see the breathing so well. One is that every time you breathe, odors come in that activates sensory neurons. The other is that it turns out that some of the sensory neurons, not a large amount, but some of them are actually also mechanosensors. So if you just breathe air that has no odors to activate them, some of them will respond. Okay? So you already have a breathing signal even without the odors. Okay. Now if you zoom in, okay, let's zoom in into this area, you can see that spikes tend to come at a certain phase of the breathing. Okay, so this is the breathing cycle. I am not sure which way here is inhale and which is exhale. Probably up is inhale, but spikes come at a certain phase. Okay, and then if you record from a different mitral cell, or when you present an odor, this phase can shift. So different mitral cells will have different phases that they like. This one likes let's say the inhalation. A different mitral cell may like this flat area. So one potential code for the fact that an odor has arrived may be not just changing the spike count, but also maybe changing the temporal pattern of spiking. And here you have an example of a neuron. Okay, so this plot shows time, and this is the phase within respiration, and each dot is one spike. And you can see that normally this neuron likes half of the phase, but then you present an odor and it likes the other half of the phase. Okay? If you look at the spike rate, okay, if you do a PSTH, everyone has seen a PSTH before? Does anyone want me to explain what a PSTH is? No. So if you look at a PSTH, the response is minimal. So most of the response for this neuron is a change in the phase preference and not a change in the number of spikes. Okay? So this is another complexity that the olfactory bulb adds.
These are M cells?
Sorry?
M cells?
Yeah, mitral cells.
Can you explain this shift in the phase?
So we know that inhibitory neurons are involved, but we don't know beyond that. At least I don't.
Do we know some distribution of phase preference within glomerulus?
Yes. Does the same? So the question, if I understand correctly, if you take different mitral cells belonging to the same glomerulus, do they like the same phase?
Yeah.
And the answer is no. So this is another. I think of all of these as heroic experiments. People recorded from what we call sister cells. So 2 cells Just remember I told you there are about 10 of them in the brain, and you don't see them. It's not that, they're not labeled with colors like I show you. So you need to record from 2 neurons accidentally that belong to the same glomerulus. And then, so at the dendrite level, they're organized. Their dendrites are all together, but then their somas are kind of spread within 300 micron range. So the N in that experiment is not huge, but that experiment shows that sister cells. I actually think I might have removed it. Yeah. They show that you can record from 2 different neurons. That if you look at spike counts, they're very similar in their responses to odors. If you look at phase shifts, they may be very different. So, which suggests really that they don't share the same inhibitory neurons. So the inhibitory network may already differentiate between mitral cells that belong to the same glomerulus. Okay. So time is interesting in olfaction, and at this point, we think that I think, other people think, that probably the best way to represent responses to odors is based on a time frame that is locked to breathing. Okay? So here you have recordings from a mitral cell. This is an awake animal. And this is the breathing signal. Okay, so 3 traces from breathing signal. And if you just do what most of us do, which is every 10 seconds or whenever you want, you ask the computer to open a valve and then odor comes out, then you happen to catch the breathing signal at different phases. Right? So starting the odor here and starting the odor here may not be the same. And if you look at the raster plot, does everyone know what we're looking at here? Or should I explain? I'll explain. This is time. Each row is 1 trial when we open the odor, and each dot is 1 spike. So we have this neuron spiking, and then the grayish area tells you when inhalation actually occurred after we opened the odor. So the odor on is always here, time 0, but then if the mouse is exhaling, then nothing goes into the nose, right?
It's for mitral cells?
Yes, this is a mitral cell. Yeah. And what you can see is that it's not really clear what the response. I mean, you can see more spikes here, but the exact structure is not clear. If you just make a PSTH out of this, you'll get kind of a ramp going up.
We said that the mitral cells don't have axons, right?
Granule cells don't have axons.
No, the mitral do have.
Yeah. Their axons go to the cortex.
Okay.
Yeah, laterally are dendrites. Okay, so now you can take the same data. Since you measured the respiration, you can now shift all the trials to be aligned not to the time the valve opened, but to the time that inhalation started. Okay? And then you get a much better structure, right? This looks like a much clearer response than this. So this is already an improvement. But it turns out that in different trials, the mouse breathes at different rates. So what they found here is that if you actually not only align to time 0, but also warp each trial, stretch it so that all trials kind of have the same length. Or in a different way, what I'm saying is that you look at when the spike occurred now as not the time since inhalation, but the phase since inhalation. Okay? Then you get the clearest response. Okay, so these are the 3 PSTHs of these. In black, you can see slow ramp, low peak, and then it goes down again slowly. In the blue, okay, so this is just aligning to inhalation. You get a larger peak, still a bit slow, going down. In the red, when you do the warping, you actually align multiple sniffs, multiple breathing cycles. So you can see multiple breathing cycles. The peak goes up, and everything is more accurate in time. And what they found is that the response is, if you do this thing, is accurate to the level of a few milliseconds across trials. So if you ask when does the neuron start responding in different trials, how similar is the timing, the jitter here is about 10 milliseconds, okay, which for many of us olfaction people was kind of surprising. We tend to think of this as a very slow kind of sense, right? Not like vision. So it turns out if you do the right alignment, it's actually pretty fast. It's not vision, but it's pretty fast.
What cells are these? Are they the sensory or the...
These are the mitral cells. We're in the olfactory bulb. Okay?
So what do you mean by the several milliseconds? Where is it condensed? The response is condensed?
So you can, there are different ways of doing this, but you can figure out for each trial where you estimate the response started. And then just ask, how different is this estimation for different trials? Okay. Okay. So let's get to the cortex. So far, what we've seen is that sensory neurons and olfactory bulb, everything seems to be pretty labeled lines, right? You record from a neuron, and it tells you what the receptor, what one receptor gene felt, right? There's importance for timing, not just the spike numbers. What happens in cortex? So there's a lot of text here. So first of all, what is the olfactory cortex? So this is, at some point, you go deeper and deeper into the brain, and asking whether a brain region is olfactory or vision or auditory becomes kind of impossible to answer and maybe not extremely interesting. But for some purpose, we need to define what we care about at this point, right? So olfactory cortex, for the way I'm going to define it, is any region that receives direct input from the olfactory bulb. Okay, so the mitral cell axons, anywhere they reach, I'm going to call olfactory cortex. Some of these regions are not cortical at all, but we in the field like to call them olfactory cortex. Okay, so what do these include? They include the piriform cortex. Okay, they include the anterior olfactory nucleus, which is, even though it's called nucleus, is at some level a cortical structure. I'm going to describe these two a little bit later. Lateral entorhinal cortex, right? It's not an olfactory region, but it receives direct olfactory bulb input, so it deals also with olfaction. Cortical amygdala, olfactory tubercle, some other regions that we're not going to discuss. Olfactory tubercle, if we get to, I'll say a little bit about, but this is basically an extension of the striatum, and current thinking is that it is involved in the valence of odors, not identifying the odor, but just linking odors to reward. Okay, kind of what people think in striatum. So of these, really the best studied is the piriform cortex, which in many papers and books, when people write olfactory cortex, they just mean this because it's the best studied. These are very, very different from each other, both in structure, in the way they receive input, where they project. But this really is the one we know the most about. And I think most people tend to think that this is where clean odor processing, for the sake of odor processing, is done. Okay? This is also true for the anterior olfactory nucleus, but we know less about it. Okay. So this is just I show this for two reasons. One, so these different names are basically the same names you had in the last slide. You can try to identify them. It doesn't matter. Piriform cortex, AON. This is olfactory epithelium, olfactory bulb, and then you have all the others. So one thing is to show you that all these regions are very interconnected. So bear in mind. The second thing that is important to see here is that many of these regions project back to the olfactory bulb. Okay, and I hope next week to get to these projections. In fact, the number of axons going from piriform cortex to the olfactory bulb is about 10 times, probably more, than 10 times the number of axons going from olfactory bulb to olfactory cortex. Okay? This is not unique to olfaction. This is true to many sensory systems. Okay. So olfactory cortex is a complicated thing, and now let's just talk about piriform, which is complicated enough. So piriform cortex is this area here. Just to orient anyone who's not oriented, this is the olfactory bulb. This is brainstem, right? Spinal cord, cerebellum, cortex. This is piriform cortex. So it's kind of somewhere here for the mouse. In a Dapi staining, in a slice, this would be piriform cortex. This, by the way, would be the AON, which we're going to talk about later, maybe. And this is the olfactory bulb. So a slice here is what we've seen before, right, with glomeruli and then the different layers of the olfactory bulb. Okay. Let's look at the circuitry in piriform cortex. So piriform cortex is what we call a paleocortex. So it's thought of as a very old version of cortex. It's 3-layered, okay, not 5 or 6 like the other sensory cortices that you've maybe learned about. And it has a bunch of different cell types. Important for us, it has pyramidal cell types, okay, that are very similar to what you'd see in hippocampus. There's different type of GABAergic neurons. And the layers are organized such that in the top, like in many other cortices, you get the input, at least the feed-forward input. Okay, so lateral olfactory tract is the input from the olfactory bulb. A little bit below, you get input from other cortical areas. Okay, so these we call association fibers. And then the output is mostly the deeper neurons, the pyramidal neurons. And this will project to many, many places. So like I showed you, to many other what we call olfactory cortical areas, but also to others, and the olfactory bulb. When am I supposed to end? A quarter to. So I have a 4 minute. No, almost. So let me just leave you with this. So the first question is really, does piriform keep the labeled lines? Okay, I'm just going to give you that piece of information. So if I record from this pyramidal neuron, D and S is for superficial and deep. We have superficial and deep pyramidal neurons. If I record from any of these, will it reflect the activation of a single receptor type? How long are we going to keep this labeled line thing, right? And there are different ways of answering this. And there are 2 anatomical experiments that have been done. And one of them is, let's label the mitral cells belonging to one glomerulus. Okay, so these 10 mitral cells, sister cells, and look at their axons. Where do they project? Do they project focally to some, all of them just go to these 10 neurons? Or do they just project all over? So this yellow thing here is the axons of these about 10 mitral cells. And you can see one thing, they don't project to just one region of olfactory cortex, but to many. And also within piriform cortex, there are many, many endings in different areas. The other way of doing this would be inject a tracer into one neuron in olfactory cortex and ask, where are these inputs in the olfactory bulb that project to this one neuron? Okay, so you can inject a rabies virus, right? Label a neuron in piriform cortex, make it green, and then make all the neurons in the bulb that project to this neuron red. And then ask, are they just all belonging to one glomerulus? So if you look at all these red dots here, what we're looking here is a section through the olfactory bulb. This is where all the neurons that project to just one single cortical neuron. So obviously they're not from one glomerulus. They're not just localized in one region. They're all over the olfactory bulb. This is a view from the top. from the side. This is kind of through the olfactory bulb. So it's all over. So a single piriform neuron will receive inputs from many glomeruli. Okay, so we lost the labeled lines. And I guess we'll continue next week.
Few announcements. First, I want to thank Danny. Danny is in the medical neurology in Hadassah. Very excellent neuroscience department, and I encourage you all to have a website to look at it. In terms of your future career, you can do PhD also in medical school, and Danny has an excellent lab there. And I know he's looking for students, so go for it if you're interested. Another thing is all of you sign to papers. Whoever needs to sign the paper is signed to a paper, great. So next week there's no, Sunday there's no class, right? Sunday
JBC, yeah.
There's no class, and next meeting is. Questions?
I'll try to come at 9. בסדר, מעניין. מה קרה? אני לא לוקח אותו עכשיו הביתה, ושכחתי זה. אז הבנתי כן, אז נסעתי להדסה. לא חניתי, ביקשתי מביביאנה שתוריד לי את זה לרמזור. אספתי אותה ובאתי, אז הייתי פה 9:20.
כן, זהו.
אבל... כן.
הם טענו כאילו שפתרו את כל המערכת בהתחלה שאין מדד אחד שאפשר שזה הוויברציה של המדד.
זהו, לכן כשאמרתי שכולם חושבים שזה קומבינטורי על קוד, אז אמרתי חוץ יש אדם אחד בעולם, שטוען שמה שקובע את הקישור זה באמת הרמות ויברציה. הוא היחיד בעולם שחושב ככה. אני לא עשיתי אף ניסוי כזה, אני לא יודע, זה אישית חזקה. אבל אנשים שניסו ללכת בעקבותיו טוענים שזה קשקוש. כן, איפה אני ראיתי את זה? אני לא חושב אם זה היה אפילו בארץ או משהו, מישהו הזכיר את זה, אבל הוא אדם בודד בעולם, זה כנראה לא נכון. כן. המאמרים?
כן.
יש שם עכשיו את השאלות, אתם רוצים עכשיו לשאלת על זה? כן. אז אני רק ארוץ את הדברים שלי ונעשה את זה בישיבה. כן.
אמרת שיש נוירונים שהם יותר מכנורצפטורים ולא מגיבים
לא, גם וגם.
גם וגם פשוט.
כן.
וגם הראת שהתבנית בזמן היא משפיעה על הפקטורים בלב, האם זה גם משפיע על הרצפטורים עצמם או שהם תמיד מגיבים ברגע שנקשר?
לא, הם יגיבו כשנקשר. תבניות נשימה ישפיעו על מתי נקשר.
כן.
הם יגיבו כשנקשר. התבניות בבלב הן תוצאה של מה שהם מקבלים מהרצפטורים, שזה תלוי נשימה, ושל תוצאה של מעגלים איניביטוריים, שיש להם דינמיקה משלהם.
אוקיי, אז זה שני דברים שמשפיעים על התבנית הזאת, גם האיניביטוריים וגם הנשימה. תודה.
בכל שיעור, אז יש עוד שיעור. בהרצאות יש הרצאה אחת, מקום 2.
בסדר, אז אני אראה.
אז אם אתה צריך, אבל אתה צריך גם לסיים את זה עם, לא יודע מה קבעתם יורם. יורם שבוע הבא מרצה?
לא,
שבוע הבא. כי אם זה, אז אולי עדיף לשמור אותו לשבוע אחרי.
אז אני אראה מה, אני צריך לראות לאן הגעתי ומה רציתי לעשות. ואני אשאל אותו אם הוא רוצה, אם אני צריך עוד זמן, אם הוא רוצה.
בקיצור, יש עוד 45 דקות, אם אתה רוצה להשתמש, תשתמש, אם לא, פשוט תן להם 45 דקות, חופש. בסביבה.
זה אני טוב.
אז הם פנו לך אלה שמקצים?
הנה, הרגע. אתה עוד חוזר לשם היום?
מה זה?
לשם. הביתה.
הייתי שם,
אני רוצה ללכת להרצאה של ההוא שהכותרת שלו היא Challenging the Boundaries of Bridge. רק בשביל להתעצבן אני רוצה ללכת.
זה היה להגיד שם גרוע, כאילו אתה מנסה למשוך אנשים טוב, זה היה
יש קטע כזה, one of the old tricks in the book, נכון? של תטען שמשהו מאוד קונטרוברסיאלי, תטען משהו מאוד קונטרוברסיאלי שהוא בעצם לא. ואתה צודק שאתה טוען שזה לא נכון, המשהו הקונטרוברסיאלי, אבל הוא מראש לא היה קונטרוברסיאלי.
כן, כן, זה מסטרומן.
זה עושה את זה לאחרונה הרבה, רפה יוסטה, שאומר, הנורון דוקטורין, שכולם חשבו שכל נוירון עושה דברים לבד, עכשיו גינו שזה רשתות של מה שחשוב. איפה היית מאז שנות ה-70 או ה-80 או משהו? טוב, יושב?
אני חושב לקפוץ לבניין עכשיו. רוצה לראות אותו?
לא, אני אחזור. רגע, איפה אנחנו יכולים לשבת? לא, אני רוצה לשבת. רגע, את זה אני צריך להשאיר פה, לא?
The most interesting part is on Tuesday. I think it would be if you do well. I asked
maybe you should say, okay, we're interested only in the multi-unit recording part. You know it's really new and you don't really know what you're doing. But do you have any logical guess about when it should come? Because we don't have much time. You know your schedule better than we, so you should suggest.
and try to start to organize to find the right outline. Okay.
Okay. Alright.
Do you want to do it on Beamer? I mean on LaTeX?
What?
Which the presentation?
What do you mean?
If we have formulas or and I might have formulas, it would be better presented.
I think that will be fine.
So we can
I
do
just
some.
don't really, I'm not familiar with that software, with the presentation.
Just
But I'm willing to try. I just
I
usually
will
work
do
with
the code,
PowerPoint.
coding part. It doesn't take too much time. But
I mean, I work with that, but for like assignments, not for presentation.
yes, the logic is what you said.
Totally fine with that. But I think until like 3 days before it should remain as a shared file.
Yeah.
And only then you can do like the animations and do like the native speaking.
Ah, yes, you want also visuals.
So I think those things should be like equally important, but it should happen.
Yeah.
Now maybe we'll organize the things, see what is the right I mean what should be the focus.
What should be what?
What subjects we want to save first and what's next and
Yes, so we can talk
the
about
order
that
of
now,
methodology.
and also what question, prepare a question, well, questionnaire. Yes, what just, we don't know what Tito is going to present, but we can think of questions we would like to talk him.
Oh,
The
this,
guy.
okay, yeah.
Am I still not pronouncing it well?